Hello again, everyone. I'm Elizabeth Schneider, Senior Global Marketing Manager here at NanoString, coming to you on the last day of AACR, the morning poster session, for another Nano News Brief. Today I'm here with Jordan Stinson. He's a graduate student at MIT, and he's going to talk to us about his work on treatment of canine soft tissue sarcomas and melanomas with intratumoral collagen-anchored IL-2 and IL-12 is safe and effective. So thank you so much for joining us, Jordan.
Of course.
So I understand that you use the nCounter analysis system for this study?
That's correct, yes.
And can you tell us a little bit about the background? What was the impetus for this?
Sure. So this is work that began by a former graduate student of our lab, Noor, where she had developed collagen-anchored cytokines as a strategy to potentiate stronger immune responses to cancers. And the sort of background and rationale for this work is we know that cytokines are very potent agents to treat cancers, but they're limited by very small therapeutic windows in terms of dose response that is efficacious versus dose response that's toxic. And so some groups have looked into intratumoral dosing as a strategy to get around this toxicity issue that's associated with IL-2 and IL-12, but inevitably, even when you deliver a cytokine into a tumor, you're still going to have leakage of that cytokine out into circulation. You can still have toxicity. So Noor's work was to develop a strategy where you could anchor cytokines to collagen-abundant tumors as a strategy to retain them in that tumor to potentiate local action without that systemic toxicity effect. And so her work in mice showed that you have this really nice relief of toxicity as a result of collagen binding, and that potentiates really strong anti-tumor immune responses in mouse models. And so to take this work a bit further, we wanted to think about using a model that's more human analogous, and so spontaneous tumors that arise in pet dogs are a great model case for that work, since these tumors develop alongside intact immune systems. They're much more heterogeneous. You can see we have different breeds of dogs that have been included in the trial, different weights, different ages, and all their names are there too. Whether that's a HIPAA violation, we're not really sure, but yeah, so it's really exciting work to sort of advance this research that Noor began in our lab to a canine model.
So are you adding some sort of collagen biomaterial to the tumor, or how are you actually doing that anchoring process?
Yeah, that's a great question. So we begin actually with the different cytokines, so IL-2 and IL-12, both fused to a canine serum albumin, shown here in gray, and that just helps increase the molecular weight and further increase the retention inside the tumor. And those are further fused to a layer domain, so this is something that's naturally going to bind to collagen. So we express these two individual proteins as single fusions, and both of them are able to be retained to collagen when delivered intratumorally.
I see. That's really, really ingenious.
Yeah.
And so what type of cancers were you guys looking at in these dog patients?
Yeah, that's a great question. So we have two different models that we've recruited patients for. The first is soft tissue sarcoma, and the second is malignant melanoma that occurs in the mouth. Those were chosen for different reasons. Obviously, recruiting patients to the trial is a challenge that we don't have to consider when we're working in mice. It's really easy to acquire as many mice as you need for research. But for pet dogs, these come to the clinic at the University of Illinois in Tim Phan's group, and present with these tumors, and so we've enrolled them in the trial. And so the melanomas help us understand efficacy of responses against these tumors that are sort of driven by IL-2 and IL-12, whereas the sarcomas, the standard of care is to surgically resect these tumors, and so that allows us to treat them, resect the tumor, and then look into what is happening mechanistically, and that's where the nCounter comes in. It allows us to profile what's taking place in the tumor to understand what's driving any sort of efficacy that we're seeing in the melanomas.
That's really cool. And so what gene expression panel did you use for nCounter?
Yeah, great question. We use the Canine IO nCounter panel, which has been really, really helpful for us to sort of work with FFPE tissues. One of the challenges that we have with this collaboration is obviously we're in Massachusetts, Illinois, University of Illinois is obviously in Illinois, and so doing the sort of exchange of materials is somewhat challenging, and it's not that we have access to take these sarcomas out and run flow cytometry, which we would do in mice, or do RNA profiling from frozen tissues. So the ability to resect these tumors surgically, go through the standard histological processing that the university has the ability to do, and then those samples can be shipped to us for RNA extraction and running on nCounter, has been really, really helpful for us to profile what is taking place inside the tumor. And so that's detailed by this panel here.
Yeah, and so what did you guys find?
Yeah, so we see with intratumoral injection of IL-2 and IL-12, this really drives robust infiltration of immune cells. And so we have different cohorts with different timings that are detailed here in the poster. But what you can sort of visually see is that in untreated tumors, you have more of an immune desert or cold phenotype, where you have very few CD3 infiltration or T cells, PAX5 for B cells, and you have some macrophage infiltration. But after treating with 2 and 12, you have this robust infiltration of T cells, B cells, in some cases, as well as macrophages. And so not only would you get that from histology, but by looking into nCounter, we can ask questions about what is actually happening with these cells that we're seeing in histology. And so nCounter allows us to profile and understand that not only are these cells arriving in the tumor, but they're also displaying signs of many effector functions that we'd hope to see for an anti-tumor phenotype. So we can profile genes associated with cytotoxicity, T cell function, antigen presentation, and NK cell function, which are all the things that you'd hope to see from an immunotherapy. And our different treatment cohorts just allow us to profile the different temporal dynamics of how this response is taking place.
Yeah. So if I remember correctly, we can do immune cell profiling with the Canine IO Panel. Did you do that at all in the study?
Yes, we have that profiled here. So the histology helps illustrate sort of more broadly what's taking place, but the cell type profiling is shown here. So not only, where histology, many of these canine markers aren't super available, the reagents are limited for canine samples as opposed to mouse samples or human samples, so we can't look at as many of these cell types. But with nCounter, we can actually profile and look at things like DCs and neutrophils and some other sort of cell type states that we don't necessarily.
You can see here clearly that the untreated sample is an immune desert, right?
Exactly.
And it looks like the cohort 1 particularly, had high levels of all these different types of immune cells.
Right, so it's great that we can use sort of the gene expression to understand what cell types are present, so beyond what we see with histology, understanding these other cell types that are present in these tumors.
Excellent. So, and tell us about the patients. Like, did you guys end up, reducing the size of the tumors? Like, did this work as a treatment?
Yeah, so on the melanoma side, these tumors are really difficult to resect because they're found in the oral cavity. It's hard to get clean margins, but they allow us a good test bed to understand how the therapy is working. So what we did was a dose escalation study, and this is still ongoing, so it's work in progress. But these dogs were recruited because they have these malignant melanomas in the mouth. They receive a standard of care 9-gray radiation dose and then 6 doses of our cytokine given every 2 weeks. And as I said, this is a dose escalation, so we began at a 1x dose, which was determined from some initial lead-in healthy beagle safety data or safety trials. But what we see is in this 1x dosing cohort, you have, by the waterfall plots, some initial response of these first 2 dogs, but ultimately they succumb to their disease. We have 1 dog in that cohort that had a really robust response. But as we increase our dosing, we get more and more near complete responses from these patients, and the overall response rate is quite favorable for intratumoral IL-2 and IL-12 treatment. And we have some dogs that are almost out to a full year since receiving their therapy.
Oh, that's so incredible. I'm sure the owners and the dogs are probably really appreciative, and not only that, but this will really help learn about how we can use this technique to treat, human patients.
Absolutely. Yeah, so this work is being moved towards human clinical trials by a company called Oncology that's actually licensed this technology for ultimate human use. And so they're very interested in sort of seeing how the canine results pan out because it helps inform how this might behave in a more heterogeneous human population. But yeah, we're very excited in sort of understanding, these 2 different models to better improve the therapy. So beyond just IL-2 and IL-12, we see that a lot of counter-regulatory responses to the immune system and activation are naturally upregulated in response to the treatment. So we're interested in taking this back to mice to understand if there's additional things that we can do to drive more robust or more durable immune responses. But yeah, I think we're really excited about how the Canine IO panel has worked for us. We're interested in sort of taking what we can also obtain from the histology data and thinking more about spatial information and how local retention, if we can stain for our protein, how that drives immune activation close to that treatment or far from that treatment, which is something that we can't necessarily obtain from histology alone or just the gene profiling.
Yeah, well, I mean, I think a next logical step would potentially be for you to use the GeoMx digital spatial profiler, and we do have the ability to do custom assays with protein. So if you have an antibody for your protein, you could, do the GeoMx digital spatial profiler. And also, we're in the process right now of getting a consortium of canine comparative oncologists together who would like to create a custom RNA assay for dogs in the context of cancer research. So we'll definitely keep you in the loop for that.
Yeah, we'd love to hear more about that, and I think it would be very beneficial for this research.
Excellent. So I have one last question.
Sure.
So I love dogs, but what about the cats out there? Are you guys going to do any research on cats? Now, I've also heard as well that compared to dogs, cats don't get as often spontaneous cancer as dogs. So I don't know if you have heard some of this from your colleagues that you work with at University of Illinois.
Yeah, no, great question. We are strongly considering it. We're working on actually making some feline versions of the proteins. They recruit patients for, I think, oral squamous cell carcinomas that can occur in cats. So we're interested in thinking about that as kind of a next step to sort of further expand on this research and test it in an additional model that is more heterogeneous than mouse models.
Yeah, I mean, I have to say, I have a friend who had a cat that died from oral squamous carcinoma. So if you can come up with a treatment for that, I'm sure she and lots of cat owners in the world would be happy. So Thank you so much,
Of course, thanks for coming by.
Pleasure. Enjoy the rest of the poster session.
Thank you so much.